I'm pretty sure you remember what a synapse is. Synapses were found at the neuromuscular junction that we learned about in the muscle chapter, but it's a location where information is transferred from a neuron to another neuron or to an effector cell, and recall effector cells could be muscles, glands, or adipose tissue. And also keep in mind that this synapse involves two neurons, if the two neurons are communicating. The presynaptic neuron and the postsynaptic neuron. There are two types of synapses: chemical, the one you're comfortable with, the neuromuscular junction, and electrical, that we'll see in the heart. So the chemical synapse is the most common type of synapse, and this relies on neurotransmitters to transfer that information from the presynaptic cell to the postsynaptic cell. Synapses that release acetylcholine, such as the neuromuscular junction, are cholinergic synapses. So we'll go over those actions that take place at that synapse. The action potential has traveled down the axon, and it has depolarized the axon terminal. Keep in mind here this ACHE, that is acetylcholine esterase. It is the enzyme that breaks down acetylcholine, and that's present in that synapse. Depolarization of the presynaptic cell, here's the presynaptic cell, opens voltage-gated calcium channels, and calcium rushes into the axon terminal. This will cause the vesicles to release acetylcholine into the synaptic cleft. This release is by way of exocytosis, and those calcium ions that had entered are quickly removed, and so that acetylcholine is no longer released. The acetylcholine will diffuse across the synaptic cleft, and it will bind to the gated sodium channel receptors on the postsynaptic membrane. Once it binds to those receptors, those chemically gated sodium channels will open, and sodium will pour into the postsynaptic cell. The more acetylcholine, the more channels open. The more channels open, the more sodium enters, and the greater depolarization. And of course, if threshold is met, then there will be an action potential generated. So once that acetylcholine has bound to the receptor, sodium has entered the cell, that acetylcholine will be broken down by acetylcholine esterase very, very quickly. Here are the events again. The action potential travels down the axon to open voltage-gated calcium channels. Calcium enters the axon terminal, and this causes the acetylcholine in the vesicles to be released into the synaptic cleft. The acetylcholine diffuses across the cleft and is bound to the sodium ion channels. Once it binds to the sodium ion channels, those channels open up and sodium enters the postsynaptic cell. Depolarization ends when acetylcholine is broken down by acetylcholine esterase. The axon terminal will reabsorb the choline from the synaptic cleft and use it to make new molecules of acetylcholine. Synaptic fatigue can occur. If there is extended stimulation, then the neurotransmitter cannot keep up with the demand, and the synapse will not function until the acetylcholine is replenished. This inability to function is called synaptic fatigue. An electrical synapse occurs in the heart, and these cells are joined, both the presynaptic and postsynaptic cells are joined together by gap junctions. And the changes in the membrane potential of one cell produce a local current in the adjacent cell, and it is just as though they are sharing a common membrane. Which of these will end synaptic fatigue? replenishing synaptic vesicles.